This is CGTN, China Global Television Network.
I'm Keeling in Cambridge, where a chemical engineer is using origami to fight antibacterial resistance.
And I'm Chinny Somara in Cambridge, where scientists are developing a sustainable form of energy by mimicking nature.
When the world gets sick, it reaches for antibiotics, or at least it used to. In recent years, our go-to treatment for bacterial infections has been overused, making them less effective.
Antibacterial resistance is one of the biggest threats to global health. 12 types of bacteria have been identified as increasingly drug resistant. But here at Cambridge University, a chemical engineer and her team are boosting the potency of molecules by delivering them directly to bacteria. And they're using the principles of an ancient art form to do it.
Origami, the Japanese word for folding paper into shapes. DNA origami is the method of folding DNA strands into nanostructures. At Cambridge University, chemical engineer Ioanna Mela had studied DNA origami for her PhD. And in 2017, she started work on her groundbreaking idea.
I figured that in order to overcome antibiotic resistance, we need to find alternative ways to target bacteria. So I thought that a clever delivery vehicle that could drive the active components, the active antibacterials, directly onto the bacterial surface would give us an advantage, or maybe to enhance or it would extend the lifetime of existing antibacterials.
DNA origami has a broad range of applications, including nanorobots, energy studies, and drug delivery. But no one was using it to combat bacteria.
So you saw a gap in the market, right?
Exactly.
Yeah, wow.
So, and we took it from there, and we have some promising results now that we're quite excited about.
To make DNA origami, you need one long DNA strand of the double helix, used as a building material to create nanostructures. DNA has 4 bases: adenine, thymine, guanine, and cytosine that bond in pairs. A always connects to T, C always connects to G. To create a self-assembling structure, scientists must design a blueprint. The blueprint involves one long strand of DNA called a scaffold and smaller staple strands that have 30 to 50 bases. The bases in the staples connect to match the bases in the scaffold, folding it into the required shape, which can be 2 or 3D. Using this technique, Ioanna and her team designed their own nanostructure in collaboration with Sugiyama Laboratory at Kyoto University. Nano refers to something so small you need a microscope to see it.
So this is our biolab, which is where the whole process of making DNA origami takes place. So I have my friend here. So this is an E. coli bacterium.
Guys, we have props. This is very exciting. I've never had a cuddly toy in a lab before.
E. coli are common in the intestines and gut. There are different strains, and while the majority are harmless, some can cause serious food poisoning and infection.
So if this is our E. coli bacterium, then we have our DNA origami nanostructures that look like this.
So this is what your nanostructures look like?
Exactly. So they are 2D, they are flat, and they have 5 holes, 5 wells. It looks a bit like the face of a 5 on a dice, right? And they have aptamers, so kind of sticky ends that allow them to specifically bind on bacterial target.
As great as post-it notes are, this diagram gives you a bit more detail. The single strands around the edge are called aptamers, and in this case have been instructed to bond only to all E. coli bacteria, so they don't kill other harmless strains. The 5 squares are wells that can hold multiple active ingredients. But for this study, Ioanna's using an enzyme found in your tears and saliva called lysozyme.
And what it does is it practically eats up the bacterial cell wall. and causing the bacterium to eventually die. And we decided to go for this to begin with because it's readily available. We can also explore other possibilities of using small molecules or existing antibiotics and see whether we observe the same effect of increasing their efficiency by delivering them in a targeted way to the bacteria of interest. The idea is that those 2D structures will bind onto the bacterial surface, and we would have multiple of those, tens or possibly hundreds per bacterium. And when the DNA origami nanostructure attaches itself to the bacterial surface, the active enzyme has then the flexibility to attack the bacterial surface and catalyze the bonds. Whether it kills it or not is a combination of how many nanostructures we can get per bacterium, but also what active component we are delivering.
So how do these two components know what shape to actually form?
So we are using a piece of software that is very handy for designing DNA origami. It's called CAD Nano.
Using Ioanna's nanostructure design, the software calculates the placement of the staples, which will bind to specific parts of the DNA scaffold once they're mixed, heated quickly, and cooled slowly to room temperature.
How many nanostructures would be in there?
So let's say if we have 20 microliters of sample, we have thousands of nanostructures in there. Yeah, so it's a pretty efficient way.
I guess it is nano.
Exactly, that is correct. That is absolutely right.
Bacteria inhabit almost every part of the body, inside and out. Ioanna's team are focusing on the outside at this early stage of their research, as the nanostructures are not stable enough to be used internally. It's hoped their method could treat the sometimes lethal infections common in hospital after surgery.
And we're hoping that by using our DNA origami loaded with antimicrobials, we can treat wounds that have become infected by bacteria.
To see the nanostructures at work, you need a super resolution microscope.
Danger laser on. Should I be concerned?
No, our lasers are actually very well contained, so we're safe.
Okay, that wasn't very reassuring.
So once we have mixed the DNA origami with the bacteria, we need to check whether they have actually attached. And we have back here our lasers.
This looks like a magic trick with all the black boxes.
It is pretty cool. So the lasers are off, so we're safe to do this now.
The lasers excite the fluorescent molecules placed in the structure, enabling Ioanna to see how successfully the nanostructures have bonded to the bacteria.
And those 4 red dots on the origami represent 4 fluorescent molecules for Alexa 647 molecules. So it's a red fluorescence that we're getting from there.
So that's just helping the microscope, doesn't have anything to do with the...
That's absolutely right. And what we end up is this type of signal. In red, we have our E. coli bacteria. And in green, we have the DNA origami.
Oh, and it's surrounded it.
Exactly, and where you can see the yellow signal is where they overlap. And we have a good amount of bacteria that are either fully covered or somehow surrounded by our DNA origami nanostructure.
Ioanna's work has attracted attention. She's come up with an idea that circumvents one of the biggest problems in antibiotic research.
The research in this field is extremely expensive. To put new drugs on the market in this area is unbelievably expensive. And because we still have many working antibiotics, companies aren't really that interested in investing that much of a risk capital into these projects. So before there really is a massive new crisis, that's not going to happen. But it's a new idea, and if these ideas are put together with those from others, perhaps then we will in the future end up with a better treatment.
Right now, what we are showing is that we can achieve the same effect, but with less amount of antibiotic when we deliver it in a targeted way. Where we are hoping we're going to be in the next few months is that we can completely kill the bacterial population using less antibiotic.
The world has been badly shaken by the spread of coronavirus. But every year, antimicrobial resistance kills around 700,000 people. Ioanna and her team are hopeful their research could be one of the new solutions so urgently needed.
Life moves pretty fast. Ideas move at the speed of sound. Technology moves at the speed of light. If you don't filter out the noise, you might miss the details. We pay attention to the details because they matter, showing you a different perspective. See the difference.
How will your world change today? What happens here? What happens there? Or what you make happen for yourself? If it matters to you, it matters to us too. Your stories are the stories that need to be told. Africa Live. Find your voice.
On the agenda with me, Stephen Cole, we look up into space. We look down into data. We look at debt. We look at politics. We look at opioids, climate change. We look at all the issues that really matter around the world. But you matter too. We want to tell the stories you want to see and hear about. Make it your agenda.
Renewable energy from the sun is a great source of electricity, but it's very difficult to store it, which is why we're so reliant on fossil fuels like petrol. But what if you could harness the power of the sun with the storability and reliability of liquid fuels? I've come to Cambridge to meet some researchers who claim they have done just that, and it's all been inspired by nature. So the idea of actually creating an artificial leaf, how true to reality is it?
So we very much look at nature and plant photosynthesis inspiration, and there's a large global field known as artificial photosynthesis that really tries to mimic these aspects. So we look how we can take some little particles known as photons from lights to drive current, to use this current to make fuels, and really this is largely inspired by natural photosynthesis.
Photosynthesis is the process by which plants take water and CO2 from the atmosphere and convert them into energy and food. The process happens inside an organelle called a chloroplast. Here, photons from sunlight provide energy by exciting electrons from chlorophyll, a light-absorbing molecule. CO2 is reduced or receives electrons, and the water becomes oxidized or loses electrons, converting them into sugars and other complex carbohydrates. Oxygen is produced as a byproduct.
The difference is mainly that we try not to make sugars, as in natural photosynthesis, but to make really energy carriers we can use as a fuel or as a chemical.
That fuel is called syngas, or synthesis gas, which consists primarily of hydrogen, carbon monoxide, and some CO2.
So syngas is produced on a very large scale globally by the petrochemical industry, and it's being made to produce hydrogen, to make fertilizers, to make methanol and hydrocarbons, and also used directly in the petrochemical industry. But the problem with syngas today is that it's being made from fossil fuels, so we are emitting a lot of carbon dioxide and deplete our fossil resources. So in our process, the idea is to really use carbon dioxide and water to make syngas sustainably.
So synthesizing photosynthesis. I can't quite believe that this can be done in a lab, but apparently I'm going to find out. Hi Virgil.
Hello.
Nice to meet you.
Nice to meet you.
So what happens here?
Well, right now we are going to interface our light absorber perovskite with the catalyst that we use for CO2 reduction.
Okay, that's a lot of complicated words.
Yes.
For me, with my understanding, leaves in their actual purest form use carbon dioxide, sunlight and water to create energy.
Yes.
What part of the process is this bit?
So basically we're mimicking natural photosynthesis by having two light absorbers which can be used to convert CO2 and water into a chemical form of energy storage. So here we will combine perovskite light absorber with a catalyst for CO2 reduction. So basically we have the perovskite light absorber here.
Perovskite is a mineral that absorbs light from the red end of the spectrum and releases electrons.
And on this other side, we have the molecular catalyst, this dark sheet. Now, in order to connect both of them, we will need to use this thin metal layer, which can melt at a low temperature, and we will heat it up and cool it down fast in order to attach the light absorber to the molecular catalyst.
Should we meld these two things together?
Yes. So, if you want to help me, could you please press the start button on the computer?
This one? So Virgil, we created this light-absorbing catalyst sandwich. What happens next?
Well, next we are going to wire it and encapsulate it, and this will basically create half of our artificial leaf.
Okay, so when you say half, why is it just half?
Because this is just one light absorber, and afterwards we will eventually contact it with the second light absorber, the bismuth vanadate.
Bismuth vanadate is another mineral that absorbs mostly blue light and releases electrons.
These are basically the two bits which will form in the end the artificial leaf. So light will come from this side. It will first pass through the bismuth vanadate light absorber and create oxygen, and then a part of the light will reach the perovskite, and on that side it will drive the reduction of CO2 into CO and water into hydrogen.
Can we see it in action?
Yes, let's go.
Yeah. But first, we need to add potassium bicarbonate to enrich water with dissolved carbon dioxide. It feels like you've combined the advantages of photovoltaic cells with the ready energy that you have with liquid fuels.
I think that's a fair statement to some extent. What we do is make the connection. We use the best photovoltaic materials and then try to connect it with our catalysts to go to fuels directly. And in this case, yes, I think we work on the transition, how to use the light directly to store the energy in the fuel.
So we're all set up. We've gone through various stages to get to this point. We've got the carbon dioxide, we've got the water, our device is immersed. All we're missing is the sunlight.
Yes. So now we can switch the light on.
Woo!
And we can already see that the bubbles start forming.
Yes. You can clearly see bubbles of hydrogen and carbon monoxide forming in these test tubes. And what's so exciting is that those are the key ingredients of liquid fuels that we may be using one day. The experiment's tiny, but the environmental impact could be huge. Even renewable energies have some kind of carbon footprint. Where does your technology sit on that spectrum?
Depends on the whole life cycle analysis. But since we source from carbon dioxide, and if we take some of this carbon dioxide and don't release it again into the atmosphere, we have overall or net taken carbon dioxide out of the atmosphere. And from this point, you can either make the fuel or you can really extract it as well. So I believe there's scope for carbon negative technologies as well on the way.
So how long will it take before we're actually pumping the liquid fuel that you've generated through this process into our cars?
So unfortunately, we are still decades away. And the reason is that our system is quite inefficient, it's quite fragile, and it's also quite costly to produce at the moment. But we're excited because the concept works. So we know in principle it's possible. The next step is, of course, to address all these limitations. And once these have been addressed, we need to scale from a little drop to a massive volume to really produce meaningful amount of fuel.
But it's not impossible, and it could really provide a solution to replacing fossil fuels then?
I think there are many possibilities to make an impact. It's transportation we have discussed, and transportation is important, and I think sustainable fuels can have clear impact in transportation. But it can also have impact in the chemical industry. So one example would be steel manufacturing. So if you can source hydrogen cleanly, then this hydrogen can be used to make steel production more sustainable. And the same would be true for agriculture. With clean hydrogen, there's also a possibility to make fertilizer more sustainable. Once we reach a situation where we can make the syngas at low cost and significant scale, then we can feed in very easily in existing processes and make these sustainable. And these go directly into the petrochemical industry. I think there's a scope for a gradual transition to have today a fossil-driven petrochemical industry and slowly convert it into a sustainable industry.
The world's currencies are more connected than ever before. The mechanisms that drive the economy are universal. Money moves markets. We explain these trends and show you how the cash in your pocket can have a wide-reaching effect. Because money makes the world go round. Global business.
Don't forget you can sign up to our daily newsletter. We bring you all the top business headlines straight to your inbox. So sign up for free at this address. CGTN. See the difference.
It's a small piece of cloth that's come between political parties and caused outrage around the world. Early on in the pandemic, health agencies and scientists couldn't agree on whether masks made a difference. Now there's growing evidence that they could be a simple solution to returning to normal life by helping us gain immunity. Dr. Monica Gandhi is a professor of medicine at the University of California, San Francisco.
We are trying to say that we're gathering one more bit of evidence that could convince people to mask, not just that it reduces transmission to others and to yourself, but that if you do happen to get the infection, that you're less likely to get severe disease.
Dr. Gandhi and her colleagues looked at 60 studies to find out what the characteristics and risk factors were for developing severe COVID-19 symptoms. And there was evidence masks may be the factor that led to the severity of the disease going down and more people being asymptomatic.
We do call it a hypothesis because number one, it's never going to get a status beyond theory. And the reason is that you can't actually randomize half a city to wearing a mask and half a city to not, and then expose people to COVID-19. It would be unethical. We have to just gather more and more observational evidence that shows the hypothesis is likely. For example, we just did an analysis with some big data scientists at MIT where we looked at 1,000 counties across the US. And it seems that right before and after mask mandates, the severity of illness from COVID-19 after mask mandates went down, as represented by hospitalizations.
The hypothesis suggests that wearing a mask could become a form of inoculation that would generate at least a short-term immunity and slow the spread of the virus, similar to how a vaccine works.
There was a study from Wayne State University in Detroit in the United States that showed that viral loads have been going down over time from April to June in people who are sick with COVID-19. Now, viral load is different than viral inoculum. We're calling the inoculum the amount you get in, and we're calling the viral load the amount that you put out. They are actually part and parcel of the same thing. They're almost two sides of the same coin, because the less that you get into your mouth and nose by masking, it seems like you have a more organized immune response and you don't get as severe disease. You don't get all these cytokines and inflammatory response and get super ill. And then the same effect makes you put out less virus, that you are able to have a better adaptive immune response, put out less of a viral load.
The team studied 3 lines of evidence: virology, epidemiology, and how countries responded.
I'll start from the virology data. It turns out that in animal studies from many, many years, 1938 was the first study that we could find, it looks like in animals, this is apparently well known because I get all these veterinary scientists writing me and saying, yes, we've always known this, that the amount of virus that you get in, the higher amount, the more sick you get. And we don't do these experiments in humans, meaning we don't give them virus and see if they get more or less sick. There's been only a couple of studies in humans like that. There's been an influenza A study published in 2015 where people actually gave wild-type influenza A to human volunteers, and the more you gave them, the more sick they got, and the less you gave them, the less sick you got. There was a study in hamsters that was published in Clinical Infectious Disease a couple of months ago where the little hamsters, they simulated masking of the hamsters. And not only were they less likely to get ill from COVID-19, meaning they were less likely to get the infection, they were less likely to get severe disease. They had very mild disease in the concept of masking.
Experiments with animals will not convince everybody that masks make a difference, but Dr. Gandhi says there's also human epidemiology research.
The example that we like to use the most is the tale of 2 cruise ships. And at the beginning, there were a lot of cruise ship outbreaks. The Diamond Princess was the famous one. It was the first one. And the rate of asymptomatic infection, people not having symptoms at all, was about 40%. And that's the current value that the CDC says is the rate of people who don't have symptoms. However, in an Argentinian cruise ship outbreak, this time they didn't let people off, but they said, okay, all of you can have masks when there was an outbreak. And they gave all the passengers masks. They gave all the staff N95 masks. The passengers got surgical masks. And after an outbreak of over 120 people, the rate of asymptomatic infection was 81%. So it was twice as much as the original outbreaks. And then the third level of data that we've looked at is country level data. This is the crudest level because there's lots of things that can be going on in countries. But in a nutshell, countries that have universally masked since the beginning of the pandemic have low rates of severe illness and death. Countries like the United States that has a more complicated relationship with masking, and it's been unevenly followed throughout the United States, has higher levels of mortality and severe illness from the infection.
President Donald Trump has only recently started to wear a mask in public and previously not always advocated their use, something he may be regretting now. Dr. Gandhi says that the early mixed messaging in public health has made it difficult to convince people that masks can save lives.
There was data from the very beginning that actually has been withdrawn now. The masks don't work. That was an unfortunate paper. And yet there's been, if you look at PubMed at this point, there's 900 articles and counting on masking in COVID-19. We're past the point that we don't think it works. This works. The hypothesis we have is around severity of disease, but masking works to prevent transmission. I think we have to sweep away the cobwebs of everything that went in the past and now start messaging appropriately.
Soon, Dr. Gandhi's team will release a report that shows how hospitalisation rates went down in the American states that advocated masks. Does that mean that, if we all put masks on, that we could get back to normal life before a vaccine appears? We wouldn't have to keep going into lockdowns and things like that?
Yes, I think that is the implication, that lockdowns, if you think about it, are a profoundly blunt instrument, and they are causing and have caused terrible economic depravity, terrible food insecurity, everything else that's coming along with closing down the economy, unemployment, all of that's going to have major health effects. We can only lock down for a short period of time. And what masks allow is the possibility of us being around each other in workplaces, in school places. And that is very hopeful news because it gives you some level of control over the pandemic while you're waiting for a vaccine.